Implantation. Male and female gametes fuse to form the zygote, which passes along the uterine tube toward the uterine cavity. After repeated mitotic divisions, the zygote is transformed into a ball of smaller cells known as a morula. Approximately 4 days after fertilization, the morula enters the uterine cavity. A fluid-filled space appears in the morula, and the morula becomes a blastocyst. By then, the zona pellucida degenerates. 6 days after fertilization, the blastocyst attaches to the compact layer of uterine endometrium. The trophoblast proliferates rapidly and differentiates into an outer multinucleated syncytiotrophoblast and an intercellular cytotrophoblast. Finger-like processes of syncytiotrophoblast penetrate the endometrium and the underlying connective tissue. The syncytiotrophoblast produces enzymes that erode the maternal tissues and enable the blastocyst to burrow through the endometrium. By the end of the first week, the blastocyst is firmly implanted and receives its nourishment from the uterine glands and capillaries. Implantation of the blastocyst is completed during the second week of development. Enzymes from the syncytiotrophoblast break down decidual cells, which provide a rich source of nutrients. The embryoblast becomes a circular bilaminar disc consisting of the epiblast, with high columnar cells, and the hypoblast, which is a thinner layer of cuboidal cells. The hypoblast forms the roof of the exothalamic cavity, also known as the primary umbilical vesicle and yolk sac, which is continuous with the exothalamic membrane. The primordium of the amniotic cavity appears in the embryoblast. It is enclosed by a thin membrane, the amnion, which is derived from the epiblast. The bilaminar embryonic disc is located between the amniotic cavity and the primary umbilical vesicle. Spaces appear in the extraembryonic mesoderm, coalesce, and form a large fluid-filled cavity known as the extraembryonic coelom. The extraembryonic mesoderm splits into 2 layers, the extraembryonic somatic mesoderm, which lines the trophoblast and covers the amnion, and the extraembryonic splanchnic mesoderm, which surrounds the umbilical vesicle. The primary umbilical vesicle decreases in size and gives rise to a smaller secondary umbilical vesicle. By the end of the second week, primary chorionic villi are formed. The extraembryonic somatic mesoderm fuses with the cytotrophoblast and syncytiotrophoblast to form the chorion, which becomes the wall of the chorionic cavity. A small circular and thickened area of endodermal cells, known as the prechordal plate, appears and serves as an organizer region. The embryo, amniotic cavity, and secondary umbilical vesicle are suspended in the chorionic cavity by the connecting stalk. Endovaginal sonography is used for measurement of the diameter and size of the chorionic cavity, which is invaluable for evaluating early embryonic development and pregnancy outcome.